moving on to the liver, the gallbladder, and the pancreas. So, although these don't have a direct contact with our inner lining or lumen space of where all the food is working, they actually are very important in our digestive system function. So, we're going to actually look at them. Okay, so starting off with the liver, so off on the right side. Yep, right side of the body. So that's what's highlighted in this bluish color. So there's right underneath the diaphragm, and that just happened to be a gallbladder, by the way. So function of the liver to secrete bile, which is going to help with our lipid digestion. That bile gets stored in here by the gallbladder. So even if you don't, if you, whatever reason, maybe get your gallbladder removed, you still can actually have function of breaking down your lipids. You might either just have to supplement, or you might have to adjust your diet just a little bit. Okay, so other job of the liver is to store glucose, amino acids, iron, and different vitamins and nutrients. So remembering all the blood of our digestive system, all of it's going to go using our hepatic portal system to the liver, and that's where the liver can then get essentially first choice of all the nutrients to help store stuff for whenever you need it later. Okay, so looking down into the liver, so our cells of our liver, called our hepatocytes, are in stacks of plates in the liver. We have our central vein that passes down in the middle, so as our blood is flowing into our liver, we have just big open sinusoids that allow for the blood to flow through them. This is also the big spaces that allow for the albumin protein that is made in the liver. So our plasma protein, that large spaces where this can be just entered into the bloodstream. So the cells of the liver are called the hepatocytes. So with our hepatocytes, again, they're in sheets of cells. They also have a brush border microvilli, so increased surface area to help with absorption. And they also filter out the blood from the stomach and intestines. So filtering out stuff so they can absorb all those nutrients coming from the stomach and intestines, as well as take in different, for example, different medicines. Sometimes the active form is because the liver has detoxed it, so converted it into a different form, because we know the body's going to do that. And so for some medicines, the active form is after the liver does its job. It also helps with detoxing, for example, alcohol. So that's why some people who are alcoholics, eventually over time, it starts damaging the liver. Okay, so job of our hepatocytes, so our liver cells. So after a meal, they're going to absorb our nutrients. So glucose, amino acids, iron, vitamins, minerals. And that just allows it for storage so that between meals, you can release them when necessary. So breaking down our glycogen so that we're releasing free glucose into the bloodstream, helping to maintain a steady blood sugar level. Remove and degrade like different hormones, because eventually you're not going to need them. So they don't need to be continuously circulating out through your digestive system, or not digestive system, circulatory system through the blood. Break down toxins, different bile pigments. So that's part of why our bile is all that green color. It's because it's breaking down that stuff, as well as breaking down your hemoglobin pigment. And different drugs. So again, some of your medications, they either get broken down in the liver, and so that's why they stop working. So that's why I have to take it multiple times a day. Or the active form is because the liver does its job. Into our bloodstream, the liver is going to secrete out albumin. So those are going to contribute to the thickness of our blood, secreting out lipoproteins. So we'll talk about them later. But in order to circulate around our triglycerides, we have to have a kind of secretory vesicle to put them in, because, again, they're hydrophobic. They're not going to like the watery, hydrophilic, liquid portion of blood, and so they have to have their own little package to put them in. We have different clotting factors, so it's helping to go into your bloodstream, this large molecule, so we need those big open sinusoids. And then angiotensin, which is going to help, tensinogen, that's a precursor that's going to help with regulating your blood pressure. Okay, so from our liver, the bile, or the products that are eventually going to help with digestion or just getting eliminated as a waste product are going to be stored into your gallbladder. So store and concentrate the bile, so it's going to help to absorb water, electrolytes, so it's more of a concentrated release of that bile into your small intestines. So what is bile? So bile is a yellowish greenish fluid with different minerals that are not needing more, cholesterol, neutral fats, phospholipids, bile pigments, bile acids. The important ones are the bile acids helping with our lipid breakdown. The rest of them are most likely going to end up as either waste products because we just don't need them, or might get absorbed a little bit, but mostly it's just to get rid of waste products because it's just extra that we didn't need. Okay, so in our bile we have bilirubin, which is breaking down of hemoglobin, so this is why your poop is brown. With our bacteria in our large intestines, we're going to break down bilirubin to our urobilinogen, again, helping to create our urine and poop color. So that's why it's the brown color that it is, unless you have a bunch of food dye in whatever you've eaten. Or a lot of iron, like your dark green leafy vegetables that can turn a little bit darker than normal, and that might freak you out, but it's just, there's all these vitamins that you just didn't, vitamins / minerals / what was I saying, iron that you just didn't absorb because there's just so much in those vegetables. Okay, so bile acids. This is the fun one. So these are a form of a steroid. So we have our cholesterol, which is our parent molecule for steroids. And with this cholesterol, we are able to make bile acids as one of the products. Those bile acids are going to help with the breakdown of our fat. So bile acids and lecithin, basically taking larger fat globules and breaking them down into emulsification droplets. Moving on to the pancreas. So the pancreas is both an endocrine gland and an exocrine gland. So it's an endocrine gland because it releases insulin and glucagon into our bloodstream. It's an exocrine gland because it releases pancreatic juice using a duct system out of the organ, but into our digestive system. So the secretion that is releasing into our small intestines is our pancreatic juice, which is water, different enzymes, zymogens, so these are precursor enzymes, sodium bicarbonate, so basically baking soda, that's going to neutralize the acid from the stomach, and then other electrolytes. So here's our pancreas again. It hides behind the stomach, so that had to be removed in order for us to see it. Okay, so release from the pancreas. So one of the zymogens or the precursor enzymes is trypsinogen. Trypsinogen gets converted to trypsin, which is going to help break down your protein. So sometimes if you're in a lab situation, you might use trypsin, and that's what it's doing. It's breaking down proteins. It's just one of the enzymes that we sometimes use in a regular lab. Other enzymes released by the pancreas, so we have amylase, so ACE enzymes to break down amyl, so our sweet or starches, carbohydrates, whatever. And again, pancreatic because it's released from the pancreas. Lipase, same thing, ACE for enzyme, LIP for lipid, so breaking down our fat, so helping to break that down. And then a ribonuclease and deoxyribonuclease to break down RNA and DNA, since, again, what we're eating is cells. Okay, moving on to the small intestine. So this is where we're going to have most of our chemical digestion. So nearly all of it, our chemical digestion and nutrient absorption, occurs in the small intestines. Now, with our small intestines, it's a bunch of tubing. It's actually very long, but the small refers to the diameter of the tubing, not the length of it. So we have 3 different regions of our small intestines. They do different jobs. So we have our duodenum, jejunum, and ileum. The duodenum is going to receive the stomach content, since it's the first section, receives the pancreatic juice and the bile, so from the pancreas and from our gallbladder, so helping to mix those all together. So in this section, this is where our stomach acid is neutralized using the bicarbonate from our pancreatic juice. So now we're back to a neutral pH inside of our small intestine. So now pepsin can't work because it works in a pH of 2. So we're going to break down that pepsin in just reuse all of those amino acids. Okay, fats are broken up, so they're emulsified, so kind of broken up, and this is also where you have physical breakdown, kind of churning, that also helps to break them up into smaller bits, as well as combining them with the bile acids to break them up into emulsification droplets. Pepsin is inactivated now that you're at a neutral pH, and your pancreatic enzymes work on chemical digestion. So remember, chemical digestion is by the enzymes. The next section of your small intestines is your jejunum. So this is where most digestion and nutrient absorption occurs. So this is where you're finally getting down to that monomer building block and into a form that can be absorbed into your small intestine cells and then moving on to our bloodstream. The last section of our small intestines, that's where you have a bunch of lymph node nodules, so just protecting our doing immune system function on whatever you've eaten. This is also where you have the ileocecal junction and valve to help control what goes from our small intestines to our large intestines. So inside your small intestines, because you are doing a lot of absorption, you have different structures that are going to increase your surface area. So we have circular folds that help to do that. We have these villi, so these kind of long finger-like extensions that are covered in microvilli. That's also going to increase the surface area, helping you to have more space for absorption. So inside these microvilli, that's where you have contact digestion. So these are going to be those final enzymes, and they're just hanging out in the microvilli, waiting for, as you are having your muscle contractions and churning and mixing up your food, and as it flows through the microvilli, then the enzymes that are inside that microvilli are going to form contact digestion. So you have to have contact into the microvilli area in order to react or essentially come in contact with these enzymes, because they don't move out of the microvilli into the open space. So stuff has to flow through the microvilli in order to make contact with these enzymes and get the final breaking down into the monomer form so that they can be absorbed. Okay, I'm going to skip some stuff that's not on the test, excuse me. So over time, the food or the chyme that has come from your stomach mixes with your different juices in your intestines. So from the pancreas, your bile, all that gets mixed using segmentation. So that mixes them all together so that the enzymes can get to the foods to start breaking them down. And then once everything has been nearly fully digested, you go from segmentation to peristalsis to move the food down through your small intestines and eventually on its way to the large intestine. So let's work on looking at how our different nutrients are digested and absorbed. So we're going to start with our carbohydrates. So for our carbohydrates, it's going to break down from whatever type of carbohydrate it is, and eventually it's going to break down into glucose, or it gets converted to glucose. So that's what eventually all of our sugar gets broken down into. So that's our monosaccharide building block that can get absorbed. So process of carbohydrate digestion, we have our salivary amylase, so salivary for saliva, ACE for enzyme, amyl for something that is sweet, so there's our carbohydrates. So that's going to help to break down the stuff in our mouth. And this one gets denatured when it comes to contact with stomach acid. So wrong pH for it to work, so it's just not going to function in the stomach. So we're going to have to start this process again when we get into the small intestines, mixing our chyme with our pancreatic amylase, so from the pancreas. And then eventually our brush border enzymes, I don't care about you knowing their names, but those brush border enzymes are going to break it down into our monosaccharide, our building blocks for our carbohydrates, so that they can be absorbed. So most of our absorbed carbohydrate sugar is glucose. And in order to bring a glucose into the cell, we have secondary active transport. So primary active transport, we have our sodium potassium pump using ATP in order to set up sodium and potassium gradients. And then on our apical surface, we have our sodium glucose co-transporter is going to bring both sodium and glucose. So here's our open space of our small intestines where our digested food is, going to bring both of them down their concentration gradient and into the cell. And then there's a glucose pump to push the glucose out of our small intestine cell and into our intercellular space, which gets picked up by our circulatory system. Another way to get glucose from our open space of our intestinal lumen through our intercellular space is with solvent drag, since glucose can dissolve in water, and so water can leak through our tight junctions between our cells. So same thing with our other monomer sugars. They can get absorbed through the small intestines and then eventually pumped out into the intercellular space, absorbed by those blood capillaries, and then our hepatic portal system is going to deliver them to the liver, so the liver can either convert them to glucose or store them in the form of glycogen. Okay, moving on to our proteins. So we have 3 different sources of amino acids. So the dietary proteins, what we've eaten, different digestive enzymes, so eventually some of our enzymes, they don't work in certain areas, and so once they become inactive, they just get broken down and we reuse those amino acids. And then sloughed off epithelial cells. So from our mouth, our esophagus, stomach, those cells that die off and break off from our digestive tract, we end up just digesting those as well. So help to recycle those amino acids. So our exogenous amino acids, the stuff that we've eaten, taken in, that's about 44 to 60 grams a day, versus our endogenous amino acids, so breaking down our own cells as well as our own enzymes, is roughly 30 grams a day. So breaking down our proteins. So we start in the stomach. So even though you might chew up stuff in your food, you don't actually have chemical digestion until you hit the stomach, where we have pepsin that works at a pH of roughly 2, so very acidic environment, which works inside the stomach. So pepsin's going to break our peptide bond, so our hydrolysis of them. So it's going to make our larger proteins into smaller peptides, and there are going to be some amino acids that just happen to get broken off. So again, pepsin works best at a pH of around 2. Then our smaller peptides get sent to our small intestine. Pepsin is inactivated because the pH there is the wrong number. So it doesn't work unless it's an acidic pH. So our pepsin is going to get broken down. Oh well. Okay, so enzymes from the pancreas are going to help to break down our small peptides into our eventual amino acids. You don't have to know them for the test. Just know these are our contact digestion enzymes. So those are the ones that are hanging out in those microvilli. So as our little peptides go through and they come in contact with these brush border enzymes, again, don't have to know the names, when they come in contact, and then they're broken down to the individual amino acids, and then those can be absorbed into the bloodstream. Okay, last one is lipids. Again, I don't really care about our nucleic acids. Whatever. It's the fat stuff. Okay, so problem with our lipid digestion. Lipids are hydrophobic. Our body is hydrophilic. There's all kinds of water. So we have to take some extra steps to break down our lipids. So starting in the mouth, we have lingual lipase. So ACE for enzyme, lip for lipids, and that's what's breaking down. Lingual referring to the tongue. So the lingual lipase that is in your saliva helps to break down your fats into smaller bits. And then we restart this process once we get into our small intestine. So not restart this process. Lingual lipase actually gets activated even more so by hydrochloric acid. You have a little bit more breaking down in your stomach, but again, you got these big old fat globules. So once you get into your small intestines, then the bile from your gallbladder, so made in the liver, stored in the gallbladder, then released into your small intestines, that's going to help to break up our larger fat globules into our emulsification droplets, which allow for the enzymes to be able to access our triglycerides and break them down. So these emulsification droplets are going to be broken down even further by, again, bile and lecithin. So essentially making a little package around these bigger fat globules into our smaller emulsification droplets. This is where pancreatic lipase can come in. So another enzyme, ACE for enzyme, lip for lipase, and it's from the pancreas. So breaking down our triglycerides into monoglycerides and free fatty acids. So down here, enzyme, breaking off of fatty acids. So we have our monoglyceride, free fatty acid, free fatty acid. So now that we've broken them down, they got to get packaged into a form that allows them to be transported because we're still in a watery hydrophilic environment. So all of our lipids are packaged from our small intestines and packaged and to move them into the small intestines into what's called a micelle droplet. So this is using our bile acids. We're packing our monoglycerides, our fatty acids, our cholesterol, and our fat-soluble vitamins. So those are A, D, E, and K. So a lot of your foods that you're like, oh, these have good vitamins. If you're eating your salad and there's no fat along with your salad, you're not absorbing those vitamins. So if you want to have your cheese on your salad, if you want to have your fatty dressing on your salad, do it, because that's the only way you're going to get some of those vitamins into your body. Okay, so our micelles, what they do is they'll take our lipids, take them into our small intestine cells, and then go back into our open space of our lumen in order to pick up more fat. So they're kind of like a grocery store, like bagger checker, whatever, like the ones that do the bagging, where they'll take your food out to the car and then go back to the grocery store and take someone else's food out to the car and go back to the grocery store. So they kind of do this repetitive going back and forth just to drop off lipids from our open space of our small intestines to our small intestine cells, so getting them across that barrier, and then going back into our open space. So these micelles are produced by the liver, stored in our gallbladder, and then released when necessary. Okay, so next step, we have dropped off our lipids from our open space or lumen of the small intestines. Micelles transport the lipids into the cells. Inside the cell, using our smooth endoplasmic reticulum, our monoglycerides and free fatty acids are resynthesized or recreated into triglycerides. So basically sticking stuff back together. This is also where we can make some more phospholipids, because that's kind of one of the jobs of the smooth ER. Then we send all of this to our Golgi complex to make another vesicle-like structure, and that is called a chylomicron. That chylomicron then takes the lipids from our small intestines and out into the intercellular space. Now, because of this big, large package, we can't actually go into the bloodstream. So these chylomicrons, these secretory vesicles, instead of going into the bloodstream, they're released by exocytosis into our intercellular space and picked up by our lymphatic system. So remembering that our little flaps of our lymphatic capillaries that allow for larger stuff to be entered in, like bacteria or like virus pathogens, whatever. So these larger packages of our lipids, they are able to be picked up also in order to eventually transport them and eventually dump them into our bloodstream. So same thing, like our rest of our lymph eventually is going to get dumped and returned back into the bloodstream. So here is all of your nutrients and what works where and who, what, and where. So like going from carbs, proteins, lipids, what's happening in the mouth, the stomach, small intestine cells themselves, and then how they're picked up. Okay, so this is for the nucleic acids. I could care less. Let's keep going. Okay, we got just a little bit. I know it's getting long, but the large intestine, there's not much to it because now you just have waste products, and then those waste are then compacted up and then eliminated. So we're going to skip through this fairly fast. Okay, so here in the large intestine, the main job of the large intestine is to compact our waste products into an easier form to get rid of them. So most of the water absorption that comes with compaction actually happens in the transverse colon. So we still again have our shapes, well, directional shapes still. So cecum is our opening, ascending, transverse, descending, sigmoid for our curve here, and then rectum and anus. There's your appendix. It's kind of a little dangly thing there. I don't know, some kind of evolutionary leftover, but oh well. It has a bunch of white blood cells, so that probably does something. Okay, so then there's your rectum and anus again. Different layering of it. Bunch of mucus that helps with keeping everything smooth, as well as fiber also helps with that, keeping stuff going. Okay, so inside our intestines, one thing we do have are our gut microbiome. So what do you call those things? Probiotics that you take. There's also other ways of taking care of those bacteria, but those are helping to that just kind of helps to refresh those and keep you having enough of these microbes. So about 800 species of bacteria. They're going to break down cellulose. So it's a plant carbohydrate that we don't have an enzyme for. Same thing with pectin. Stuff that we don't have an enzyme for, those bacteria actually help us to break it down. Some of it's just to break down fiber, and that way it can line our digestive system, but also just to break down stuff that we just don't have a enzyme for. Also can help with creating vitamins B and K, which again are just vital and necessary, and so they also help doing that job. Unfortunately, part of their function is as they release their waste product, they create a gas which is called flatus. So part of that comes out your rear end is because of the bacteria doing their job, but most of it's because of swallowed air as you have ingested food. Okay, so most of what the large intestine does, it's your absorption of water. So that happens in your transverse colon where you're reabsorbing water and electrolytes that just happen to be dissolved in the water. And that helps to compact up your feces. And eventually, as you're pushing the feces through, you have your hostile contractions that kind of push stuff toward your rectum and anus, and then your mass movements, that's actually going to trigger that kind of, oh, I gotta go feeling. So helping to just kind of keep stuff moving forward. So they actually start when you put food into your stomach, and some of it also goes to the duodenum, so that first section of your small intestine. So basically as in, hey, move out the way, we got more food coming. So just kind of, okay, emptying out the space because you got new stuff coming in. Okay, so then again, defecating, there is a bit of a reflex because you filled up the rectum, and so it's like, okay, we got to get rid of what's there. So it's a reflex to get stuff that's out. So controlling this, again, we do have a sphincter muscle that is a skeletal muscle, so that's why we are able to learn how to control it, but we also can forget how to control that, and that's why Grandpa wears Depends. Okay, so defecation only occurs if you are relaxing that external sphincter and therefore allowing for it to move out. And I think, yep, there we go. Okay, so that is the end of whatever that chapter is, chapter 25. So go ahead and answer your study guide questions. And, answer study guide. I just went blank. Oh, ask me questions if you need to, because that's what I'm here for. See you for the next chapter.